Welcome to the Science of Ageless Living. I'm Dr. Ethan Hausman-Marquis, doctor scientist, and the only peptide clinic physician in London with a published body of work specifically in this field. This is the Ageless Top 20 Peptides series. Every episode covers one compound, the real biochemistry, the honest evidence base, what we know, what we don't, and what the regulatory picture actually looks like. No hype, no discount codes, just the science. Today, Clotho. If you've never heard of it, that's about to change. And if you have, you've almost certainly heard a version that's been significantly oversimplified. In 1997, a Japanese researcher named Makoto Kuroe was working at the University of Texas Southwestern Medical Center. His team had inserted a foreign gene into mice, a routine enough procedure in the lab. But something unexpected happened. The mice aged rapidly, not just a little, dramatically. Within weeks of birth, these mice developed osteoporosis, arteriosclerosis, skin atrophy, muscle wasting, and cognitive decline. They died at a fraction of their expected lifespan. Every hallmark of premature aging compressed into a few months. When Kuroe's team identified the gene that had been disrupted, they found it was a previously unknown protein, and because of what it appeared to do, govern aging itself, sKlotho named it after Clotho, the Greek fate who spins the thread of life. They called it Clotho. The inverse experiment was equally striking. Mice engineered to overexpress Clotho lived 20 to 30% longer than wild-type controls. Not healthier for the same duration, longer, extended lifespan in a mammalian model. That's the kind of result that gets a field's attention. And since 1997, Clotho has become one of the most intensively studied proteins in aging biology, over 4,000 published studies, a clear correlation with human longevity in observational data, and a mechanistic picture that, the more we understand it, becomes more rather than less interesting. So let's go through it properly. What Clotho actually is, what it does at the molecular level, what the human data shows, and what the honest picture looks like for anyone in this field. First, the basics. Because Clotho is not a peptide in the conventional sense. It's a protein, a glycoprotein to be precise. And like many proteins, the form matters enormously. There are two primary forms of Clotho, and conflating them is one of the most common errors in how this compound is discussed. The first is the full-length transmembrane version, a single-pass transmembrane protein, meaning it sits in the cell membrane with one end facing out and one end facing in. It has a molecular weight of approximately 130 kilodaltons. In this form, Clotho functions primarily as a co-receptor. It combines with a fibroblast growth factor receptor, specifically FGFR1C or FGFR3C, to form a high-affinity receptor complex for a hormone called FGF23. Without Clotho, FGF23 cannot efficiently bind its receptor. With it, the binding is highly specific and potent. FGF23 is produced by bone cells, and its job is to regulate phosphate and vitamin D metabolism via the kidney. When this signaling axis is disrupted, which happens when Clotho is absent, you get abnormal mineral metabolism, soft tissue calcification, and accelerated vascular aging. Sound familiar? It's what the Clotho-deficient mice showed. This transmembrane form is expressed most abundantly in the distal tubule of the kidney, in the brain's choroid plexus, and in the parathyroid gland. The second form is where it gets particularly interesting for longevity biology. The extracellular domain of transmembrane Clotho can be cleaved, shed, by metalloproteinases called ADAM10 and ADAM17. The resulting fragment, roughly 68 kilodaltons, is released into the bloodstream and acts as a circulating hormone. This is shed Clotho, sKlotho, and it's the form that circulates systemically, reaches distant tissues, and mediates most of the pleiotropic longevity-associated effects we associate with the protein. sKlotho doesn't require a cell membrane to act. It travels in blood, cerebrospinal fluid, and urine, and it has been shown to interact with multiple receptor systems in tissues throughout the body. When researchers talk about Clotho as a longevity biomarker, they are typically measuring circulating sKlotho, And when they talk about Clotho levels declining with age, which they do dramatically, it's the circulating form they're measuring. And this brings me to what I think is the most clinically significant finding in Clotho biology. The age-related decline in circulating Clotho levels is substantial and well documented in humans. Studies have shown approximately a 40% reduction in circulating sKlotho between the ages of 40 and 80. Not a gradual drift, a significant, measurable decline. Multiple large observational studies have associated lower Clotho levels with higher all-cause mortality, faster cognitive decline, greater cardiovascular risk, and accelerated biological aging markers. Whether that association is causal, whether restoring Clotho levels would reverse those outcomes, is the central open question in this field, and I'll come back to it honestly. The reason Clotho has attracted such sustained research interest is that it appears to interface with multiple hallmarks of aging simultaneously, not through a single pathway, but through several distinct mechanisms. Let me go through the major ones. The first is IGF-1 and insulin signaling suppression. One of the most conserved findings in aging biology, from yeast to nematodes to mammals, is that reducing insulin and IGF-1 signaling extends lifespan. The pathway runs IGF-1 receptor activation, PI3K, AKT, mTOR, and it's the same pathway implicated in rapamycin's longevity effects. sKlotho suppresses this pathway. It inhibits IGF-1 receptor and insulin receptor signaling, reducing downstream PI3K and AKT activation. In doing so, it appears to activate longevity pathways that are conserved across species. This is mechanistically significant because it means Clotho may be functioning as an endogenous version of the kind of signaling suppression that some of the most compelling longevity interventions in model organisms achieve pharmacologically. The second mechanism is WNT signaling suppression. WNT signaling drives tissue development and cell proliferation. It's essential during development, but in adult tissues, excessive WNT activity is associated with fibrosis, cancer promotion, and stem cell exhaustion, all hallmarks of aging. sKlotho has been shown to directly bind and sequester WNT ligands, preventing them from activating their receptors and reducing downstream beta-catenin nuclear translocation. The antifibrotic implications are particularly studied. In kidney tissue, cardiac tissue, and lung tissue, Clotho deficiency correlates with increased fibrosis, and restoration of Clotho in animal models attenuates it. Third, NRF2 and the antioxidant program. NRF2 is a transcription factor that controls the cellular antioxidant response. Its targets include heme oxygenase 1, NQO1, superoxide dismutase, and glutathione peroxidase, the major enzymatic antioxidant defenses. Clotho activates NRF2, and the result is upregulation of the cellular antioxidant capacity, reduced ROS burden, and attenuation of oxidative stress-mediated damage. This has been most thoroughly studied in kidney and cardiovascular tissue, where oxidative stress is a major driver of age-related dysfunction. Fourth, TGF-beta and fibrosis suppression. TGF-beta is one of the primary drivers of tissue fibrosis. It signals through SMAD2 and SMAD3 to activate fibroblasts and drive extracellular matrix deposition. Clotho inhibits TGF-beta receptor signaling, reducing SMAD2/3 phosphorylation and downstream fibrotic gene expression. In the kidney, this is one of the most well-replicated findings in the Clotho literature. Clotho deficiency accelerates renal fibrosis, and Clotho supplementation in animal models attenuates it. And fifth, ion channel regulation. In the kidney, sKlotho has been shown to directly modify TRPV5, a transient receptor potential channel involved in renal calcium reabsorption by cleaving its end-linked glycans. This stabilizes the channel at the apical membrane and enhances calcium reabsorption. This mechanism is more directly tied to mineral metabolism than to longevity per se, but it illustrates the breadth of Clotho's biological reach. It's not acting through a single receptor. It's modifying the glycan landscape of membrane proteins, directly affecting their function. So to summarize the mechanistic picture, Clotho appears to suppress two major pro-aging signaling axes, IGF, 1-TON, insulin, and WNT, while simultaneously activating the antioxidant program and inhibiting fibrosis. That's a compelling combination, and it explains why the phenotype of Clotho deficiency looks so much like accelerated aging. Now let's talk about what the human data actually shows, because this is where I need to be careful, and where a lot of the Clotho narrative overclaims. The human evidence falls into roughly three categories: observational associations, genetic studies, and a very small number of interventional findings. The observational data is the most abundant. Multiple large prospective cohort studies have measured circulating Clotho levels and followed participants for years, looking at health outcomes. The findings are consistent. Lower Clotho is associated with faster cognitive decline, higher cardiovascular mortality, greater all-cause mortality, reduced physical performance, and worse kidney function. These associations hold even after adjusting for age, kidney function, and other confounders. A 2017 study in the Journal of Gerontology followed over 3,000 adults for 12 years and found that individuals in the lowest Clotho quartile had significantly higher all-cause mortality than those in the highest quartile. I want to be clear, these are associations. Observational data cannot establish causality. It's possible that Clotho is a marker of biological health rather than a driver of it. The protein may decline because of aging rather than causing aging. That said, the consistency of the associations across multiple cohorts and multiple outcome measures is notable. The genetic data is more informative. Genome-wide association studies have identified variants in the KL gene that are associated with longevity. Perhaps most striking, the G allele of a specific mitochondrial SNP that affects Clotho expression is enriched in centenarian families across multiple independent cohorts. A functional variant called KLVS, a haplotype carrying 2 amino acid substitutions, has been studied extensively. Heterozygous carriers show better cognitive performance in some cohorts, improved cardiovascular markers in others, and in some studies, extended longevity. Homozygous carriers of the variant, interestingly, show worse outcomes in several studies. A non-linear dose response that complicates the picture, and then the interventional data. Here is where I must be completely direct with you. There are no completed phase 2 or phase 3 human clinical trials of exogenous Clotho administration. There is no approved Clotho product. The interventional human data is essentially non-existent at this point. What exists is early phase safety work and exploratory studies. A small number of phase 1 trials have begun examining safety and pharmacokinetics of recombinant Clotho administration. But efficacy data in humans is not yet available. Everything we know about what happens when you increase Clotho in living systems comes from animal models. And while those models are compelling, the lifespan extension, the cognitive preservation, the anti-fibrotic effects, the history of longevity biology is littered with compounds that worked beautifully in mice and failed to translate. Resveratrol, telomerase activators, NAD precursors in their early optimism. The translation problem is real. I say this not to dismiss Clotho. The biology is genuinely compelling. But because you deserve an honest account of where the evidence actually stands. I want to spend a moment on the cognitive data specifically because it's where some of the most exciting recent work has been done. In 2014, a landmark paper in Cell Reports showed that overexpression of Clotho in aged mice improved spatial learning and memory, synaptic plasticity, and long-term potentiation in the hippocampus. The effects were not modest. Aged mice with elevated Clotho performed comparably to young mice on spatial memory tasks. The proposed mechanism involves enhanced GluN2B subunit function at NMDA receptors, a synaptic plasticity mechanism, and increased myelination through Clotho's effects on FGF receptor signaling in oligodendrocytes. In humans, the observational picture is consistent with the animal data. Lower Clotho levels are associated with poorer cognitive performance across multiple domains, and a 2015 study in Annals of Clinical and Translational Neurology found that the KLVS heterozygous variant, associated with higher Clotho expression, correlated with better cognitive performance in adults over 60, independent of APOE4 status. That last point is worth emphasizing. APOE4 is the strongest genetic risk factor for Alzheimer's disease. The fact that Clotho's cognitive associations appear independent of APOE status suggests it's operating through a different mechanism, not just modifying the APOE pathway. Whether a therapeutic Clotho strategy could translate these findings into clinical benefit for human cognitive aging is the question. We don't know yet, but the biology is coherent and the associations are strong enough that this is one of the more actively pursued areas in the broader neurodegeneration research space. Given that Clotho declines with age and lower levels are associated with worse outcomes, the obvious question is, can you raise it? And if so, how? Exercise is the most robustly supported intervention. Multiple studies have shown that both aerobic exercise and resistance training increase circulating Clotho levels in humans. A 2020 meta-analysis in the Journal of Sport and Health Science pooled data from 16 studies and found significant increases in circulating Clotho following exercise interventions, with the effect most pronounced for moderate intensity aerobic exercise sustained over 12 weeks or more. The magnitude of increase is around 10 to 20% in several studies, though it varies considerably. Vitamin D is also worth mentioning. Clotho expression in the kidney appears to be regulated by the vitamin D receptor. Studies in cell culture and animal models suggest that vitamin D signaling upregulates Clotho expression, and several human observational studies have found positive correlations between vitamin D status and Clotho levels. This is one of the more mechanistically coherent relationships in the Clotho literature. Though interventional data is limited, animal studies have consistently shown that caloric restriction increases Clotho expression, likely through AMPK activation and mTOR suppression, overlapping with the pathways through which Clotho itself acts. Human data is more limited, but some studies of intermittent fasting protocols have found modest increases in circulating Clotho. And then there's the interventional frontier. Several biotech companies are developing recombinant Clotho proteins and Clotho gene therapy approaches. Early phase 1 safety data is beginning to emerge. One research group has published preclinical data showing that a single systemic injection of recombinant Clotho in aged mice produces sustained cognitive and physical improvements lasting weeks beyond the protein's plasma half-life. This has been interpreted as evidence of lasting epigenetic or synaptic changes rather than simple pharmacokinetic effects. Whether this translates to humans and at what dose and frequency is entirely unknown, but it's one of the reasons Clotho remains near the top of the longevity biology research agenda. So, where does this leave Clotho in 2026? The honest assessment. The biology is among the most compelling in longevity science. The mechanistic picture, suppressing IGF-1 and WNT signaling, activating NRF2, inhibiting TGF-beta-driven fibrosis, touches multiple hallmarks of aging simultaneously. The animal data is consistent and reproducible across labs. The observational human data is robust across multiple cohorts and outcome measures. The genetic data provides a degree of causal evidence that purely observational studies cannot. What we don't have is interventional human evidence. We don't know whether exogenous Clotho is safe in humans at effective doses. We don't know what the effective dose is. We don't know whether the animal findings translate. We don't have a licensed product. We don't even have completed phase 2 trial data. Anyone selling you Clotho today, whether that's a supplement claiming to increase Clotho or an injectable formulation without phase 2 data, is operating significantly ahead of the evidence. What we can say is this: Clotho is one of the most scientifically credible longevity targets currently under investigation. The basic science is strong, the observational evidence is consistent, the interventional frontier is genuinely exciting. And if the current wave of phase 1 and phase 2 trials produces positive results over the next 3 to 5 years, Clotho could move very quickly from research compound to clinical practice. In the meantime, the evidence-based strategies for supporting Clotho, regular moderate intensity aerobic exercise, optimized vitamin D status, and maintaining healthy metabolic function are worth pursuing on their own merits regardless of the Clotho data. That's Clotho, named after the fate who spins the thread of life, first discovered in 1997 through a mouse with a disrupted gene. Now one of the most intensively studied proteins in human longevity biology, with an evidence base that is genuinely compelling at the preclinical level and still developing at the clinical level. In the next episode, we'll be covering penadica arginate, a newer arginine-enriched analog of BPC-157 with a scientifically coherent mechanistic rationale and an evidence base that is frankly even earlier than Clotho's. We'll do it the same way: what it is, what the proposed mechanisms are, what the evidence actually shows, and what the honest picture looks like. If this series is useful to you, share it with someone who's navigating this space. This field deserves more rigorous public discussion and less noise. I'm Dr. Ethan Hausman-Marquis. This is the Science of Ageless Living.